Okay, and welcome back. So in this video, we're going to continue our look at heredity and how traits are passed on. This time, though, we're going to be looking at traits that are what we refer to as sex-linked. So these are traits that are carried on either the X or the Y chromosome. So they're particularly linked to those two chromosomes that determine the sex of an individual. So these are our X and Y chromosomes that we're talking about in this case. So, as we've talked about before, humans have 46 chromosomes in body cells, okay, and 44 of these are autosomes or chromosomes that are specifically for the body, and 2 of these are sex chromosomes, so our XX or our XY. So if a genetic trait occurs on an autosome, then males and females are equally affected, because if it's on chromosome 1 through 22, males and females get those in the same kind of ratios. However, and we can use our monohybrid cross, so remember these ones, A, we can do what we call a monohybrid cross to work out the ratio of recessive or dominant. Okay, so this is a monohybrid cross, and we use this for complete dominance. Okay, so where we have a recessive and dominant characteristics are our two options for our alleles. However, it's different if that trait occurs on a sex chromosome. Okay, so remembering that these are our X or Y chromosomes. If the trait is carried on either one of these, then males and females may be affected differently, because females have the XX and males have the XY. Then if it's carried on an X, then the ratio of the alleles can be different for females and males. We refer to these as sex-linked crosses, okay, because they're linked to the X and Y chromosomes. So what this means is that the conditions that are often sex-linked are often linked to the X chromosome, so they're more common in males than females. This is because the Y chromosome, this one here, is actually smaller than the X, so it carries fewer genes. This is the X here. We can see that it's actually much larger. So in the males, we have XY, so this would be a male combination here. So many of the genes that are on the X chromosome don't actually appear on the Ys. So males will only have one allele, so they only have one version of that gene. Okay, so a single gene on the X chromosome can control the phenotype of the male then, because they don't have that two allele combination, which would give them another chance of having a non-affected allele. So there's greater than 50 diseases and conditions that are caused by recessive genes on the X chromosome. Okay, so this is where we would have the recessive version being carried on the X chromosome. So in this case, if it was carrying a recessive version on the X, there is no other option on the Y, so this is the only form it would get, so the trait would be shown. Okay, these include things like color blindness, some forms of hemophilia, albinism, and a few others. I think albinism is. So if we look at sex-linked, okay, this means that they are linked to the X or Y chromosome. It's not always the X, but it quite often is. So because males are X and Y and females are XX, if a linked condition is dominant, males and females are equally affected. Okay, because if it's dominant, then it will show in the female, and if it's dominant, it's the only version the male gets, so they will show it as well. If the condition is X-linked recessive, then males are much more likely to be affected because they get only one copy. So if they get the recessive one, it will automatically be the phenotype, because there's no dominant one to make the recessive version silent. Okay, and if it's Y-linked, then it can only affect males because females don't have, they have no Y chromosome, so they can't be affected by it at all. Okay, because males have X and Y, then they can be affected by X-linked conditions, but females can't be affected by Y-linked conditions. So, if we have a look, females have 2 X chromosomes and males have only 1. So this means that if a female has a recessive condition on the X chromosome, then and a dominant allele on the other X chromosome, this will mask the condition. So they will be a carrier, but they won't actually have the condition. Okay, so they can carry it. They wouldn't be affected by it, but they can pass it on. If a male has a recessive condition on the X chromosome, he will have the condition because there is no chance of an alternative allele being there to mask it. So 2 common examples of these are hemophilia and colorblindness. So when we complete Punnett squares for these, we need to show clearly which sex chromosomes we're talking about. So we need to specify if it is an X or Y. Okay, so we use the same structure as what we have before. A capital is going to be a dominant trait, and a lowercase letter is going to be a recessive trait. Okay, so in this case, females can have 2 possible combinations. Well, actually 3. 2 for not showing the recessive trait, so we can have a homozygous dominant. You will notice in this case we need to show the sex chromosome, and then the dominant allele is shown as a superscript. Okay, or the recessive allele is shown as a superscript with the smaller lowercase here. So this one is going to be homozygous dominant. This one is heterozygous dominant. The phenotype for both of these will be unaffected. Okay, they will not have the condition. Then in the case of this one, we can see we have 2 copies of the recessive allele, and this is the only way that a female can be affected by the disorder. Okay, whereas with a male, they can have the dominant trait, which means that they are unaffected. But as soon as they have a single copy of the recessive trait, they will be affected by the disorder. So males cannot be a carrier. They will either be affected or unaffected. Females can be unaffected, affected, or a carrier of the trait. So if they're a carrier, they carry the allele for it, but they're not affected by it. So a female without the trait, showing dominant unaffected alleles in both chromosomes. Female who is a carrier but will not show the trait. A female with the trait will have the 2 recessive alleles. Male without and male with. These are all the possible genotypes that we can have for a sex-linked recessive trait linked to the X chromosome. Okay, so this is showing it in a diagram form. So in this case, this would be an unaffected father. Okay, a carrier mother because they carry the recessive form as well. An unaffected son, in this case the blue is showing us unaffected. An unaffected daughter because both versions of the allele are unaffected. A carrier daughter because she's received this gamete from the mother and has an unaffected one as well, and then an affected son because he has received the affected allele from the carrier mother. So as we said, this can occur in diseases such as hemophilia. So hemophilia is a blood disorder where blood doesn't clot properly. So normally what happens is we start to bleed, the vessels constrict, platelets form a plug, and then we get a clotting factor forming, which stops us from bleeding. If you have a bleeding disorder, bleeding can start, the vessels will constrict, but the platelet plug isn't finished and the clot doesn't form. So the person can continue to bleed, which is quite dangerous. So if we were to look at this in terms of the genotypes, we can say that hemophilia, because it is a recessive X-linked disorder. Okay, so we're going to give it a small letter H, and then the dominant trait, which is non-hemophilia, will be a capital H. So a female who does not have hemophilia and is not a carrier will be X with the double H on both of her X chromosomes. A female who is a carrier will be a X with a capital H on one chromosome and the lowercase h on the second chromosome. The rarer combination is where we end up with a female that has two recessive alleles, one on each X chromosome. This female will be affected by hemophilia. So a man without hemophilia can only have one genotype, that is the X with the non-hemophilia allele. And then a male with hemophilia will have the X chromosome with the hemophilia recessive allele on the X chromosome, remembering that the Y doesn't provide us with any genetic information for hemophilia. So when we go to solve these problems, we're going to read the question carefully, making note of what is dominant or recessive, remembering that the dominant gets the capital letter and the recessive gets the lowercase. If it is autosomal or sex-linked, okay, if it's autosomal, then we're just going to use our monohybrid crosses. If it's sex-linked, then we're going to remember we need to show the X and Y chromosomes. We'll determine the genotypes of the parents, remembering this is the allele combination, so our big A, little a, or XH. Draw up the punnett square with the allele combinations. Okay. Determine the genotype and phenotype proportion, and then comment on the answer to the question if it's asked. So I know this is going on a little bit long, so we'll just do one example and then we'll talk about this more in class. So in this case it says hemophilia is a recessive sex-linked on X. So we know that it's going to be a lowercase on X. We need to determine the chances of the children of a father who has hemophilia and an unaffected mother. Okay, so if the father has hemophilia, they must be this genotype, the X with the hemophilia allele, and a Y. The mother, on the other hand, has two possibilities. They can be a dominant and recessive, or they can be two dominants. So this is, if we were highlighting this question in our test, recessive sex-linked is important on X. The father has hemophilia and then an unaffected mother. Okay, so the mother can be heterozygous or homozygous, but there is only one option for the father. So there are two types of punnett squares that we can have. We can have this one here where we have the X homozygous mother, and this is the punnett square that we get for this. So homozygous mother, then our affected father, which would give us a carrier daughter. This would also be a carrier daughter. This would be an unaffected male in our phenotype and a affected male in our phenotype. Okay, so we can see this, that we would have these two together, which is 2 out of 4, which is 40%. Without hemophilia and female, we would have 25%, so 1 out of 4, males without hemophilia, and 1 out of 4, 25% males who are hemophiliacs. The other Punnett square possibility that we have is where the mother is a carrier. So in this case, we're not homozygous, we're heterozygous, where the mother is a carrier. This will result in some different phenotypic results that we get. In this case, we have a carrier female, one of each. We have an affected female, so this female would carry, would express the disease. We have an unaffected, remembering that boys can't be carriers, male, and an affected male. So in this case, we have a 25% chance of a female without the trait, 25% chance of female with the trait, 25% chance of a male without the trait, and 25% chance of a male with the trait. So we can see how the genotype of the mother can have a large effect on the genotypes, the phenotypes of the offspring, given that it is an X-linked disorder. So just a note on our ratios. So when we're talking about ratios, the phenotype is the trait that is expressed, the physical appearance of that. The genotype is our allele combination. And when we talk about the possibility, we give it as a percentage, or we can give it as a ratio. So this would be a 1:2 ratio, or a half. Okay, so you need to mention the sex of the offspring as well as the percentage for sex linked. But when we're talking complete dominance for autosomal traits, you don't need to mention the gender. So we have largely completed this, and we'll be working on this again in class. And then you have your textbook questions as well. And we'll leave it there.